Welcome to the webinar about scavenging various impurities and scaling up this purification process using the E-PAK technology. So you have been working in the lab for the last few years developing a new molecule, and you are now ready to move it up to the commercial scale. We wish it would be as straightforward as possible. However, it is not always the case when moving up and can take many ways to get there. Would it be nice that you could take everything and transport them to a pilot or larger system? What if the same condition you have in your lab can easily be replicated on a larger scale and still yield the same purity? With E-PAK, it can be, at least when it comes to the purification of metal or organic impurities. Let's take a look at the steps of the scale-up process and see where we can simplify things for you. Hi, my name is William Sommer. I'm a product manager at Cycle for the E-PAK product line. And today, we're going to talk about scaling up your purification using the E-PAK technology. Let's go over the scale-up process and how we can simplify the purification. The three main steps are choosing the right scavenger, the lab scale optimization, and the scale-up, with the end goal being a straightforward process, starting with the screening and selection of the scavenger. The first step is where your purification starts. And the first question is, what are you trying to remove? Metals, catalysts, organic impurities? Based on what you are trying to remove, that will help narrow down the best scavenger to use. The benefits of using scavenger gives you scavenging versatility, where you can remove a wide range of metal species in various oxidation states and ligands in any solvents. It can be used in different formats, whether it's batch as a powder or fixed bed as a cartridge. You get no API contamination, with no to minimal extractables and leachables from the scavenger, with very high scavenger purity. And you get high selectivity, with total recovery of the API in most cases. Here at Cycle, we offer the C-Amethyst product line, which is a whole line of scavengers that are efficient for a wide range of impurities. This chart and other helpful information are available in the catalog or on the website, and is a great source of information to help guide the starting steps in selecting and screening the best one for your purification needs. Many of the solvents that are offered in bulk are also offered in the cartridge format. You can see in this slide that many of the popular silica-based scavengers are included, as well as activated carbon solvents. As you may have noticed in the previous two charts, certain impurities or metals may have more than one possible scavenger that can be used. This is why a screening test is important. Depending on the chemistry of your reaction, scavenging efficiencies can vary compared to the previous results. Parameters affecting efficiency include the nature of the metal catalyst, the nature of the ligand used, the solvent used, the structure of the API of the molecule trying to purify, and the presence of any residual reagents or byproducts. After an initial screening, the next step is lab scale optimization. The lab scale optimization allows you to adjust several parameters: the number of equivalents, subsequent treatments, the reaction time, the temperature, the nature of the solvent, or the mixing rate. The graph on the right is a good representation of this step. In the example here, for the scavenging of palladium acetate with C-amethystyl and DMF, by adjusting both the number of equivalents and the temperature, scavenging went from 75% after 1 hour with 1 equivalent at room temperature to 100% in less than 5 minutes with 4 equivalent at 80 degrees. In order to determine how much scavengers is needed, two types of calculations are possible: the molar equivalent method and the weight method. For sake of time, I would direct you to our explainer video available on YouTube that goes into more detail and step-by-step on how to use them. Okay, so we found the right scavenger and did the lab scale optimization. Now, most people would jump straight to scaling up. Instead of going directly for a larger setup, the trick is to transfer and apply the result to a lab scale E-PAK. By adding this tiny extra step, it will allow time saving, since everything can be linearly scaled up with ease to treat up to several hundred kilograms of solution. Applying the previous parameters you optimized to the cartridge system will be pretty straightforward. And that includes the cartridge size, the treatment method, and the contact times that will be dictated by the flow rate. To go from bulk powder format to the appropriate cartridge size, this table shows you the corresponding amount of both silica and carbon-based solvent. By transferring to E-PAK, you are now ready for an easy scale-up. The beauty of the E-PAK technology is that you can use the lab scale parameters that you optimize, and you will be able to transfer all of those to the larger sizes, whether there's a pilot or commercial scale, which in this video you see is 100 centimeter housing. These include flow rate, the type of solvent, the amount you will be using, and the impurity you're trying to remove. As you can see in this video, you have your solution going through the E-PAK cartridges and coming out clean, being transferred into another reactor. This is where the time from step 3 of transfer from bulk to the cartridge system pays off, as linear extrapolation will not be possible, resulting in a straightforward scale-up. And of course, the condition must remain constant. The scavenger to impurity ratio, the residence time, the temperature, the solvent being used, and the contaminant and compound levels you're trying to remove. To walk you through the linear extrapolation, we put together this table, which have all the different cartridge size available. You can see that we have the scale-up factor, which gives you the multiplying factor to go from the smallest cartridge all the way to the biggest one. The mass of silica and of carbon for each size cartridges. The bed volume, which corresponds to the total volume of the cartridge without the volume of the hole. The recommended flow rate, which is for residence time of 2.5 minutes, which experimentally proved to be the best time to get the best scavenging. The flow rate range that you can use each cartridge at for residence time between 1 to 20 minutes. And the conditioning, which corresponds to 3 tanks volume being needed to prepare the cartridge before you're ready for your purification. To illustrate this linearity, we used 5 different cartridges with 5 different sizes. The smallest one's 5 by 1 centimeters, all the way to 16.5 by 100 centimeters, doing scavenging of palladium acetate. And you can see that the scavenging efficiency and the kinetics are pretty much the same from the first cycle all the way to the 7th one. Now let's look at it with a practical example. In this case, the scavenging of copper and palladium after sonogashare coupling. For the first step, we're going to do a cilia carb and cilia methane screening, followed by parameters optimization, then transfer it to the E-PAK, and finally scanning it up. In this reaction, we're taking an aniline to do a sonogashare coupling using palladium tetrakis and copper iodine. This reaction was taken from Smith et al. From an OPRD paper from 2019. For this reaction, we're using a 20-liter reactor with 2 kilograms of the aniline, 220 grams of palladium tetrakis, and 36 grams of copper iodine. At the end of the reaction, we end up with 8,360 ppm of palladium and 895 ppm of copper that we're trying to remove. The first 2 steps, the bulk screening and the optimization, are done with the cilia carb. And we found that cilia carb VW, 50% weight in toluene at 22 degrees for 4 hours, allow us to go from 8,360 ppm of palladium to 769 ppm of palladium. And for copper, able to get down to less than 5 ppm. We did the same thing with cilia methane, and we show that cilia methane DMT, 30% weight in ethyl state at 22 degrees for 4 hours, gave us less than 10 ppm of palladium. Now we're ready to transfer it to the E-PAK. Using a cilia carb VW 50% weight corresponding to a 5 by 1 centimeter cartridge in toluene at 22 degrees for 3 hours. With a flow rate of 8 milliliters per minute, we were able to get down to 752 ppm for palladium and for copper at 5 ppm. For cilia methane, we went with 25% weight using a 5x1 centimeter cartridge in ethyl state, 22 degrees for 4 hours. With a flow rate of 8 milliliters per minute, we're able to get down to 12 ppm of palladium. To showcase the linearity, we transferred those conditions onto 4 different cartridges, with the smallest one being the 5x1 centimeters, all the way to our pilot scale cartridge size, the 16.5 by 12.5 centimeters. Using the different scale-up factors that were showcased earlier, you can see that we were able to get API recovery in the 90% and scavenging efficiency of up to 99%. It is important to note here that the solution to purify stayed at the same concentration for the API and the impurities. As you can see in this practical example, the scaling up of a purification process using E-PAK is seamless. You go through the same steps with scavenging selection, optimization of parameters, and adding the E-PAK, which allow you to do easy scale up. Don't forget that E-PAKs comes in many sizes in a variety of solvents for a wide range of impurities. There should always be an E-PAK solution for your purification process. A little bit about us. We have been purification experts for the last 25 years. We have an expert R&D team that is supporting product development with the highest quality. Products are available worldwide, and our customer support is always happy to help. For our contact info and more information about E-PAK and other purification products, go to our website, citycycle.com. Here you can find case studies, application note, and other relevant information. Thank you for listening.